Science in Pajamas. Hey guys, welcome back. So, today we're going to talk about mutations. This was one of the last things we started talking about in class, and I want to talk a little bit about it again today. So, I know it's been a little bit. Hold on one moment. Just a bit. There we go. So, it's been a little bit. When we left off, we were talking about how mutations are changes in the DNA. And oftentimes, when you change the DNA, that means that you can change, no, go lay down. You can change the mRNA, the messenger RNA, which means you can change amino acids in the amino acid sequence, which means you can change the protein. And that's why we look at these, especially since we just talked about protein synthesis. Now, there are three main classifications of mutations that we're going to talk about. We mentioned these in our notes. The first is a point mutation. A point mutation is when one single base is exchanged for another. Now, there's a few different effects that this can have. So here I have an mRNA sequence. We have the protein sequence, the amino acid. So again, this is mRNA. So AUG methionine, UUU, phenylalanine, GCA, alanine, UGG, tryptophan, AAG, lysine, ACG, threonine, CCC, proline, UAA. Stop. So when we have the DNA that matches or is complementary to this mRNA, this is the amino acid sequence we would get. But what if there's a change to the original DNA? So the original DNA of, let's say, GCA. I'm just going to pull up my handy dandy little chart. So we know that GCA is alanine. But what if, so we know the mRNA is GCA, and that gives us the amino acid alanine. Now remember, the DNA is complementary to the original DNA. Did I say that right? The mRNA is complementary to the original DNA. So that means the DNA would have had to been complementary to GCA, would be CGT. Now, maybe during replication, an error occurred, and this T gets replaced with a C. No, we'll just put this on its own separate line. So we have CGC now. That means that the mRNA would be GCG. Now, if we look at our amino acid chart, GCG would still give us the exact same amino acid, alanine. We call this a silent mutation. It's still a change in the original DNA, and that led a change to the mRNA, but there is no change to the amino acid, which means there won't be any change to the actual protein sequence. However, that's not the only kind of point mutation we can have. Let's see. What would be a good one? Let's see. Yep, okay. So AAG. AAG is lysine. That means, again, complementary DNA would have had to been TTC. Now, what if there is a mutation and instead of, let's see, so instead of that, we have TTA. So the last base got changed from a cytosine to an adenine. That would make our mRNA, AAU. And if we look at our handy dandy little chart, that would give us asparagine. So we have one amino acid swap for another. Now when we have the amino acids in their sequence, the number, the exact placement of each of the amino acids is what will determine how the protein is folded and what kind of function it'll have. So switching out one base for another could cause an amino acid to be switched, and that could have a minor effect, it could have a big effect, because depending on which amino acid it is, it could have a large effect on how the protein is supposed to fold. It could completely change its folding. So it all depends, again, lots and lots of unknowns, but that is a possibility. So we do have this kind of point mutation. Now there's also some, still working on point mutations, but this time I'm going to go at this fourth codon, UGG. So we know that UGG codes for tryptophan, and the complementary DNA sequence that would go along with that is ACC. Well, maybe we have a point mutation that is going to affect the second base. So we end up with, so we have ATC. So we're switching up that second base. That will lead us to UAG, and UAG is a stop codon. That means that's going to have a huge effect on the protein, because if we put a stop codon here, instead of at the end where it normally would be, none of these amino acids would be added. So it's going to shorten the amino acid chain, which means that the protein itself is going to be drastically different. It's going to be missing whole chunks. So those are point mutations. Yeah. Now we're going to talk about frameshift mutations. Frameshift mutations are ones where the reading frame of the DNA is essentially just how we look at how we read it in terms of codons. And a frameshift mutation is when you either add or subtract bases. So extra bases are gained or some of the bases are lost, and that causes all the others to shift. So I'm just going to do a very short one for this. Let's see, we have AUG, CCC. I'm going to do like a 6. Let's see. UCC. And we have methionine. Proline. UCC is serine. Then we have GCA, which is alanine. Then we have AGG, which is arginine. And then we have UGA, which is stop. So let's say that is a particular sequence. In a frameshift mutation, again, you either add or subtract bases, so you gain or lose them. Let's say maybe we end up losing this first C. So now that means that the next codon after methionine is going to be CCU, which is, luckily for us, still proline. And then we have CCG, which is also proline. And then we have CAA, CAA, which is glutamine. And then we have GGU, which is glycine, and then we have GNA. They don't code for anything. You need 3 bases to form a codon. If you don't have 3 bases like this poor little GNA, that's not going to code for anything. So look at that. Most of the actual amino acids have now been changed. And a lot of times kids are confused about how that works. So I like to use sentences involving 3 words to show how it doesn't make sense. So let's say, Jim saw the dog and cat run. I choose 3-letter words to symbolize the 3-letter codons. So if we get rid of the S is gone, it gets lost. Then we're left with Jim, AWT, HED, OGA, NDC, ATR, UN. So we went from a sentence that made sense to a sentence that did not. And that's what we're talking about with frameshift mutations. And the same could happen if we add letters. So in this case, maybe we didn't lose the S, maybe we put a F in there. So we added an F. So now we have Jim, SAF, WTH, EDO, GAN, DCA, TRU, and a lonely little N at the end. Again, we went from something that made sense to something that did not make sense. I'm trying to get that out of the glare. And that's what we're talking about with frameshift mutations. Now, the last one we didn't get into too much detail on. We just kind of introduced them. Now, these all affected one gene. But then the third type, which are chromosomal mutations, they would affect more than one gene. They would actually affect all the genes on a chromosome. So let's say here's our chromosome. And we have genes A, B, C, D, E. So that's the order of the genes. Now, in a chromosomal mutation, something happens and that causes the genes to change their order, whether you end up with some that are lost. So maybe we end up with A, D, E, or sorry, not lost, changed. And B, C are down there. They're not in the right place. Could be some are lost. So maybe we have just C left up there, and then D and E down there. Could have extra pieces added on or extra copies. So you could have A, B, B, C, and then D, E at the bottom. So all this would affect the order of the genes, which would then have an effect on how / when / even if they got expressed. So I just wanted to remind you about some of the mutations we were talking about. Probably tomorrow I'll do a video on asexual reproduction, the very, very last thing we talked about. And that's pretty much what we're going to call it for today. So I hope everyone's doing well, and I will talk to you later. All right, bye-bye. Stay safe.